Student, we will continue competition number 4.2, and today's topic of discussion is cholesterol biosynthesis, site and organ for cholesterol synthesis, precursors, key enzyme, product formed from cholesterol, regulatory step, metabolic fat and excretion of cholesterol. Cholesterol biosynthesis takes place in all nucleated cells of the body, but the main organs are liver, intestine and skin, and out of these three, liver is the major organ for cholesterol biosynthesis. In this organ, cholesterol is partly synthesized in cytoplasm and partly in the endoplasmic reticulum. That means few steps of cholesterol biosynthesis pathway occurs in cytoplasma and few occurs in endoplasmic reticulum. Cholesterol biosynthesis takes place in post-meal state of the body. Substrate required for cholesterol biosynthesis is acetyl-CoA, and the source of substrate is glucose. In the post-meal state, glucose is available in all cells. By glycolysis, that glucose is converted into pyruvate in cytoplasm. Then that pyruvate enters into the mitochondria, and with the help of enzyme pyruvate dehydrogenase get converted into acetyl-CoA. So that acetyl-CoA, which is formed in the mitochondria, act as a substrate for cholesterol biosynthesis. Therefore, we have to transport that acetyl-CoA from mitochondria to the cytoplasm. So this is the transport of acetyl-CoA from mitochondria to cytoplasm. So here glucose in the cytoplasma is shown, which is converted into pyruvate by glycolysis. Pyruvate enters into the mitochondria, forms acetyl-CoA. Now this acetyl-CoA has to be transported to cytoplasmic compartment for synthesis of cholesterol, because cholesterol synthesis starts in the cytoplasm. Now this acetyl-CoA is impermeable by inner mitochondrial membrane, therefore it combines with oxaloacetate to form a compound called as citric acid or citrate. The enzyme required for this step is citrate synthase. So this is the first step of Krebs cycle. When this citrate is formed in large quantity, then this citrate crosses the inner mitochondrial membrane and enters into the cytosol. In the cytosol, the citrate is broken down with the help of enzyme ATP citrate lyase. Here energy is required for this enzyme, which is supplied by ATP, and this enzyme breaks the citrate into acetyl-CoA and oxaloacetate. Now this oxaloacetate is reduced to malate by malic enzymes, and then this malate enters into the mitochondria, and in the mitochondria it gets again oxidized to oxaloacetate. So in this way the acetyl-CoA is transported to the cytosol. Now when the substrate acetyl-CoA is available in the cytosol, the cholesterol biosynthesis pathway starts. So it is a lengthy pathway, so we will see various steps of this pathway. So step 1 is called as condensation. So in this step, two molecules of acetyl-CoA with two carbon combines or condenses to form acetoacetyl-CoA. The enzyme required for this step is acetoacetyl-CoA synthase. One coenzyme A is removed, and this step occurs in cytoplasm. Next step is synthesis of HMG-CoA. HMG-CoA long form beta hydroxy beta methyl glutaryl-CoA. So one molecule of acetoacetyl-CoA combines with another molecule of acetyl-CoA. So 4 carbon plus 2 carbon combines to form 6 carbon beta hydroxy beta methyl glutaryl-CoA or HMG-CoA. The enzyme required for this step is HMG-CoA synthase. This step also occurs in cytoplasm. Step 3 is synthesis of mevalonate. This is the rate limiting step of this pathway. The HMG-CoA which is formed in the previous step get reduced with the help of electron donated by NADPH. Here NADPH is the donor, not NADH. So after reduction, mevalonate with 6 carbon is formed. The enzyme required for this step is called as HMG-CoA reductase. So this is the rate limiting enzyme of this pathway, or it is allosterically regulated. This step also occurs in cytoplasm. Now step 4 is production of isopentenyl pyrophosphate. In this step, 6 carbon mevalonate undergoes decarboxylation to form 5 carbon isopentenyl pyrophosphate. before that, this mevalonate undergoes 3 successive phosphorylation by adding 3 high energy phosphate bond from ATP to form mevalonate 5 phosphate, mevalonate 5 pyrophosphate and mevalonate 3 phospho 5 pyrophosphate. Now this compound is unstable, which undergoes decarboxylation to give one carbon dioxide molecule and forms isopentenyl pyrophosphate. All these steps occurs in cytoplasm. Now from this step, there is no necessary to write down the enzymes of this pathway. Now fifth step is again condensation. Now it occurs in 4 different sub-steps. So first is isomerization. So isopentenyl pyrophosphate with 5 carbon undergoes isomerization to form 5 carbon dimethyl allyl pyrophosphate. The enzyme is isomerase. This step occurs in cytoplasm. Now one molecule of isopentenyl pyrophosphate and one molecule of dimethyl allyl pyrophosphate get condensed to form 10 carbon geranyl pyrophosphate. The enzyme is synthase. This step also occurs in cytoplasm. In the third condensation, 10 carbon geranyl phosphate condenses with 5 carbon isopentenyl pyrophosphate to form farnesyl pyrophosphate. The enzyme is farnesyl pyrophosphate synthase. Now 15 carbon farnesyl pyrophosphate is formed in cytoplasm. Up to this step, the cholesterol biosynthesis pathway occurs in cytoplasm. Now this farnesyl pyrophosphate with 15 carbon enters into the endoplasmic reticulum and the remaining steps occurs in the endoplasmic reticulum. When this farnesyl pyrophosphate enters into the endoplasmic reticulum, the 2 molecules of farnesyl pyrophosphate with 15 carbon each get condensed to form a squalene. 30 carbon squalene is formed. Here again the hydrogen are required, which are donated by NADPH. So enzyme is squalene synthase. So this step occurs in endoplasmic reticulum. Next step is formation of lanosterol. It occurs in 2 steps. First, squalene undergoes oxidation by addition of oxygen atom or OH group into the squalene to form squalene 2,3 epoxide. Here again the reducing equivalent is NADPH. So this squalene 2,3 epoxide is formed by enzyme called as monooxygenase. So monooxygenase is a class of enzyme which add one atom of oxygen from a molecule to one compound and another atom is added to the other compound. Here out of 2 oxygen atom, one is added to the squalene to form squalene 2,3 epoxide and another oxygen is added to form a water. So therefore they are called as monooxygenases. Now squalene 2,3 epoxide is a unstable compound which undergoes cyclization to form cyclic structure with 30 carbon called as lanosterol. So this lanosterol is the first compound with steroid ring formed in the body. It has 30 carbons. So enzyme here is called as cyclase. So this is the structure of lanosterol with 13 carbon. If you compare the structure of lanosterol and cholesterol, then you found 3 major differences. Lanosterol have 30 carbon, whereas cholesterol have 27 carbons. So these 3 extra carbons are attached, 2 in the form of methyl group at 4th position and 1 in the form of methyl group at 14th carbon in the lanosterol. That means cholesterol has 27 carbon, lanosterol 30 carbon. So these 3 extra carbons are present in the form of methyl group and 3 methyl groups are present in the lanosterols. Out of these 3, 2 are present at 4th carbon and 1 is present at 14th carbon. So this is the first difference. Second, if you compare the structure of cholesterol and lanosterol, then you found that there are two unsaturated bonds are present in lanosterol between 8-9 position and 24 and 25th position, whereas in cholesterol there is only one unsaturated bond, and which is present between 8th and 9th carbon. So this is the difference between lanosterol and cholesterol. Now in the remaining steps we will convert the lanosterol into cholesterol. So first, the 3 carbon atoms which are extra in lanosterol are in the form of methyl group are removed, and then 27 carbon zymosterol is formed. This step is also taking place in endoplasmic reticulum. Now in this step, zymosterol has a double bond between 8th and 9th carbon. So that double bond is shifted to 5th and 6th carbon. So the compound now is called as desmosterol. So this step also occurs in endoplasmic reticulum. In the last step, the double bond which is present between 24th and 25th carbon atom of desmosterol gets saturated with the help of reducing equivalent supplied by NADPH, and 27 carbon cholesterol is formed. So this step also takes place in endoplasmic reticulum. So in this way the cholesterol biosynthesis occurs in the body. Now we will study the regulation of cholesterol biosynthesis. Hormonal regulation first. So insulin stimulates the pathway because cholesterol biosynthesis occurs in post-meal state, whereas epinephrine and glucagon inhibit the cholesterol biosynthesis pathway. Next type of regulation is allosteric regulation. Here the allosteric enzyme is HMG-CoA reductase enzyme. So inhibitor of HMG-CoA reductase is cholesterol, which is the end product of that pathway, and the substrate is HMG-CoA. So it is shown like this. Here the allosteric enzyme HMG-CoA reductase is there. It has two sides, active site and allosteric site. So at active site the substrate HMG-CoA will bind, and at allosteric site the inhibitor cholesterol will bind. Cholesterol is the end product of the pathway. So if the concentration of substrate is more, then substrate will bind at the active site and pathway progresses. If the concentration of inhibitor cholesterol is more, then cholesterol binds at the allosteric site, brings the conformational change in the active site of the enzyme, so that the substrate will not bind at the active site and pathway stops. Next factor which regulate the availability of substrate is availability of acetyl-CoA pool. So when acetyl-CoA is available in large quantity, then it stimulates the cholesterol biosynthesis pathway, and this occurs particularly after high carbohydrate diet and diabetes mellitus. In diabetes mellitus, the blood glucose level is high, so liver will take more and more glucose from the glucose, because liver is not dependent on insulin for uptake of glucose. Hence liver will take more glucose and it will synthesize more and more acetyl-CoA. Next type of regulation is regulation at gene level. Here also the end product, that is cholesterol, act as a inhibitor of transcription of gene synthesizing HMG-CoA reductase. Now this is a artificial regulation. Here again the drug statins are used to decrease the blood cholesterol level, because cholesterol may leads to various diseases like atherosclerosis. So we have to control the blood cholesterol level. To control that, a group of drug called as statins are given to a patient. How they will act? They inhibit the enzyme HMG-CoA reductase, but by competitive inhibition. Here there is a competition between substrate and inhibitor to bind at the active site of the enzyme HMG-CoA reductase. As we are giving the drugs in large quantity, the concentration of inhibitor drugs is more, so they will succeed to bind at the active site and block the enzyme by competitive inhibition. Now we will see what is the fate of cholesterol. So average Indian diet contains 0.4 gram of cholesterol, and adult body will synthesize 1 gram of cholesterol. So daily 1.4 gram of cholesterol is added in the body. Out of this, 500 milligram of is converted into neutral sterol and excreted through the bile. Now, daily bile acid, 500 milligram of cholesterol is converted into bile acid in the liver. These bile acids are required for digestion and absorption of fat. Small amount of cholesterol is also converted into vitamin D in the skin. In the skin, a precursor of vitamin D called as 7-dehydrocholesterol is present. When ultraviolet rays of the sun falls on that 7-dehydrocholesterol, it gets converted into cholecalciferol or vitamin D. Now, next fate is synthesis of adrenocortical hormones. Here, hormones like glucocorticoids, mineralocorticoids, cortisol and sex steroids are synthesized by the cortex of the adrenal gland, and these hormones are also derived from cholesterol. Next is synthesis of sex hormones like testosterone and estrogen. So these hormones are synthesized by testes and ovaries. Next fate is small amount of free cholesterol is also present in the cell membrane. Due to presence of free cholesterol in the cell membrane, there is increase in the fluidity of the cell membrane, and this fluidity is important for semi-permeable function of the cell membrane. So in this way, this is the fate of cholesterol. Cholesterol is not catabolized in the body. It is excreted in the form of neutral sterols and small amount of bile acids in the stools. So these are the structure of bile acids. Role of polyunsaturated fatty acid in cholesterol metabolism. Polyunsaturated fatty acids binds at the hydroxyl group at third position of the cholesterol to form cholesterol esters, and after that binding, that cholesterol is transported by the HDL to the liver for its excretion. So polyunsaturated fatty acid decreases the cholesterol level, whereas the cholesterol binds with saturated fatty acid get deposited into the peripheral tissue. Therefore, it is advised that saturated fatty acid increases the cholesterol level.